So my talk will be about ocular drug targeting technologies. And first I will maybe just introduce you that where I am located at the moment. You can see here is Finland, and Kuopio is located about a little bit over 300 kilometers from Helsinki towards the northeast. And as you can see, we are surrounded by lakes. And this is the campus where the School of Pharmacy is located. And the whole university is, I think, the second biggest in Finland. And it has about 16,000 students and 2,750 employees. First, I will give a brief introduction about retinal diseases and different treatment options which are available. Then I will go more into detail about aptamers, which are the molecules that we are using to target these ocular tissues, and how these aptamers are selected. And then after that, I will just show you some recent results which we have obtained with these aptamers. And as you can see, not every animal has only 2 eyes. For example, spiders, they can have actually up to 8 eyes. Okay, but let's first get started with this visual impairment and blindness. About 30% of the world population is suffering from some sort of impairment. However, at least half of it, they have causes which actually could be prevented, or they could be at least addressed. And you can see that most of the problems are related to just simply using eyeglasses. And then, of course, cataract, which is like the lens which is in the eye, it gets opaque. It's not anymore transparent. There are many eye conditions that can cause visual impairment, and they are mentioned here. And in the first world countries, the most common causes of blindness are macular degeneration and glaucoma. And as you can see, the projected number of cases is going to increase, and this is mostly due to population aging. So all these diseases are coming in in the later part of life. And so you can see that it affects actually many millions of people. So it's a very important problem. And now I just give a brief description about macular degeneration, which is the leading cause of blindness in developed countries. And it has a prevalence of about 25% for people over 60 years old. And what happens when you get macular degeneration, you can see from this picture, is that the central vision is getting lost, so that people can still see in the periphery of their visual field, but they can't anymore see in the middle. And of course, this causes a lot of problems because these people can't anymore read or drive a car, and they will eventually lose their independence. There are 2 types of AMD. There is the wet AMD, which affects about 10% of people. And in this wet AMD, there are some new blood vessels which are starting to grow into the retina. And this type of AMD is treated by injection inside the vitreous of some anti-VEGF antibodies, which kind of block the growth of this blood vessel. But then there is about 90% of cases suffer from dry AMD, where basically some cells inside the retina start to atrophize, they start to die. And for this type of disease, there is not yet any treatment available. Dry AMD occurs over several years, so it can develop for many, many years before the patients eventually have some problems with vision. But with wet AMD, it can occur very quickly, so in the matters of months. And as I mentioned, this AMD or the wet AMD is treated with these injections, which have to be applied directly inside the eye. They are very widely used since there are many patients suffering from this disease. So there is been more than 20 million injections per year. However, these injections cannot be used with all types of drugs. They can only be used with very potent drugs, which have a wide therapeutic index. The injections have to be repeated every 1 to 2 months for the rest of the patient's life. So you can see that it's also quite of a burden for the patient to go and get these injections. And also it's, of course, quite costly because it has to be done in a hospital. And overall, it's quite a burden for the healthcare. And of course, although these injections are generally considered to be safe, there are some risks which are very severe because these risks by themselves, they can cause blindness. And such risks are, for example, endophthalmitis, which is inflammation inside the eye, or then vitreous hemorrhage, which means there is some bleeding inside the vitreous, or then the retina can detach from the back of the eye. So the reason why these drugs cannot be given in less invasive way is that in the eye, there are different types of barriers which prevent the drugs from reaching the back of the eye. Here you can see a section of an eye. This is the anterior part where we have this transparent cornea, the lens. Here there is the vitreous, which is like a gel-like substance, transparent. And then in the back we have the retina and the blood vessels of the choroid. And then the sclera is this kind of very thick tissue, which is keeping the eye in its shape. And so in the front of the eye, the cornea is a very tight epithelium, which is formed, which is kept together by tight junctions. And even the best, most lipophilic drugs, which can cross this barrier, they will have a very low bioavailability. So less than 5% of the drug can go to the aqueous chamber. And no drug can basically reach the back of the eye once it's given topically. Then here there is the conjunctiva, which is a little bit more loose than the cornea, but it also has tight junctions. And it is possible to deliver drugs subconjunctivally. You can inject up to 0.5 milliliters, and then it could reach the back of the eye by crossing the sclera and then the choroid. And then, sorry, okay. And then finally there is the retina, which is the layer which is, for example, affected by AMD. And as you can see, it's formed by many different types of cells. And here we have the blood vessels of the choroid, and then there is this retinal pigment epithelium, which is also a monolayer of cells, which is also joined by tight junctions, and it will prevent the free flow of drugs from the blood to the retina. So these are the main barriers which are present in the eye and which will make the delivery of drugs to the retina quite challenging. And so that's why until now, the drugs have to be administered directly into the vitreous if you want to treat the back of the eye. And then I was mentioning that these intravitreal injections are not good for all types of drugs. For example, if you have small molecular drugs, they will be eliminated from the eye, both from the posterior route and also from the anterior route. The posterior route is much more efficient, so it will eliminate the drugs very quickly. And so the half-life of small molecular drugs in vitreous is only 1 to 10 hours. So if you need to treat a chronic condition, it's not feasible to give these injections basically daily. In the case of proteins, so these antibodies which are used in the clinics, Nowadays, the elimination occurs only via the anterior route because the back of the eye has these tight junctions which will prevent antibodies from escaping from there. So in that case, the half-life of these antibodies is about 5 to 10 days, and that's why it is possible to administer them every 1 to 2 months. So basically, summing up what the challenges are for this retinal drug delivery is that first we have these ocular barriers, which are preventing drugs from reaching the retina and causing low bioavailability. And then we also have a fast elimination of drugs from the retina. And these drugs have a short residence time there, so the therapeutic effect is quite challenging to achieve. Of course, many people study alternative administration routes, which would be less invasive and more pleasant for the patient. One option is to have these eye drops, which are actually currently used to treat the anterior diseases of the eye. The benefits of using eye drops is that they are non-invasive, patient-friendly. But then on the other hand, until now, it is not possible to reach the back of the eye or to reach therapeutic concentrations in the back of the eye if you deliver the drug as eye drops. Another option is to have this subconjunctival delivery, which is also less invasive than intravitreal injections because the retina is quite far, so the risk of damaging the retina is quite minimal. And the benefit over this corneal or topical administration is that it will bypass the conjunctiva, and then drugs can hopefully reach the back of the eye. However, also there are barriers which have to be crossed, and until now there are no therapies which are delivered or administered as subconjunctival injection for retinal diseases. And the final option is to just have an IV injection, which is also very easy to administer. However, you will have to overcome these barriers again. And then, of course, because the drug will mostly go to other parts in the body, you may need to use very high dose of the drug, which can also cause systemic side effects. And also the drug residence time in the retina might be too brief to achieve a therapeutic effect. So here I would like now to a little bit switch to these nucleic acid aptamers. And these are the molecules which we are planning to use to target the different types of drugs to the retina, and hopefully allow the administration of drug via less invasive administration routes. So what are these aptamers? Aptamers are short single-stranded DNA or RNA, which can bind very specifically to a certain target. Usually it's a protein. So you can see here, for example, you have this HIV reverse transcriptase in gray. And then we have this orange aptamer, which is bound to a pocket into the protein. These aptamers are usually about 6 to 30 kilodaltons. And they were first isolated in the 90s by several groups. And how they bind these proteins is that they form a very defined three-dimensional structure, which is responsible to recognize the target. Aptamers are selected by a method called SELEX, which is an iterative selection procedure for aptamer generation. And all the SELEX protocols have the same, basically the same way where these aptamers are produced. First, the aptamers are administered and they will bind to the specific protein. They will also, of course, if you give, for example, these aptamers to an animal, they will partition to all types of tissues. Then you will need to recover the tissues which you are interested in and recover the aptamers which have bound to these tissues. And then you need to amplify the aptamers which have bound to the tissues and eventually inject them again for a second round of selection. And this is how it looks. This is actually a protein-based Celex. First, you have this initial library of aptamers. And we are using RNA aptamers, and our initial library has 10 to the 15 different sequences. So the sequences will eventually bind, or at least some of the sequences will bind to the protein of interest. Then you need to recover those sequences which have bound, amplify them, and then once they are amplified, you give them for the second round of selection. And of course, eventually you need to then sequence those sequences in order to know what are the aptamers which are actually binding to the protein. There are different types of Celex. There is this protein Celex, which it was just presented before. And in this case, proteins are bound to a matrix, like for example, a membrane. And this is the simplest way to perform this Celex. It's easy to perform. And these conditions in which the selection takes place can be, of course, optimized. And the targets can be either intracellular or extracellular proteins. However, there are some disadvantages. If you don't know what is your protein of interest, of course, you can't go with protein Celex. And then there are some proteins which simply can't be immobilized to some membrane, or then if they are just functional in their native conformation, then it's not possible to do this protein Celex. So the next step, or a little bit more complicated Celex, is the cell-based Celex. And here basically you have a positive population of cells. For example, you have cancer cells, and then you have a negative cell population, or like, for example, these healthy cells. And then you give the aptamer library to this positive cells and to the negative cells to get rid of these unspecific aptamers. Here you can, of course, select aptamers which bind to unknown proteins, and also which bind to proteins which are in its native conformation, because they are usually on the cell surface. The problem is that nucleic acids tend to bind to dead cells, so you need to really make sure when you do the selection that your cells are in a healthy state, and you need to evaluate how many of the cells are dead. And then finally, there is in vivo Celex, which is what we are actually using. And the aptamer library in this case is directly administered to an animal. And the reason why we use this in vivo Celex is because we want to select for aptamers which can penetrate these ocular barriers. So this can't be really done in any other way. And also we want to have aptamers which are working in vivo. And this is, of course, more complex and more time consuming than these previous two selection methods. Here you can see again the specificity of the aptamers will increase for these in vivo Celex, but of course then you have the other side of the coin that it's much more time consuming. And usually you say that you get what you select for. So sometimes you can find some aptamers which bind for proteins in this protein-based Celex, but then if you try them in vivo, they might not work. Here there are some aptamers which are currently in the clinic or in the clinical trials. And actually the only aptamer which has been approved so far by the FDA and the EMA is this Macugen, which is actually used for AMD. And I will talk a little bit more about this Macugen in the next slides. here you can see that it binds quite tightly to the target protein. And then there are some other aptamers which are in clinical trials, also for AMD. Then there are some which are meant to be used for coagulation diseases. You can see again that these aptamers bind with very high affinity. They can be compared to antibodies. Some of them, the clinical trials have been terminated because of toxicity. I think especially this one, actually the cause of the toxicity was then found to be this polyethylene glycol, which was attached to the aptamer. So it was not directly related to the aptamer. And here we have some also which are in trials for cancer and inflammation. So this is this pegaptamide or Macugen, which is used for AMD. It is a RNA aptamer that targets VEGF 165 in wet AMD. So it works quite similarly to these antibodies, that it binds VEGF and then it blocks it. However, this is much more specific, so it will only bind to VEGF 165. And that is actually the problem of this aptamer, because actually it was later found out that due to this high specificity, it is less effective than these antibodies. And actually now it is, although it is still like approved for AMD, it is not anymore used because these antibodies are more effective. As you can see, also this one is pegylated, and the reason for that is so that it would be like larger and stay in the eye for a longer time. And the backbone has been modified, of course, because if you have like native RNA, it will be removed quite quickly by enzymes. It's not stable, so you need to modify the backbone. And this was injected quite similarly to antibodies every 1 to 2 months. So some challenges, of course, with these aptamer-based therapies is that there can be this nuclease degradation. So the in vivo half-life is less than 10 minutes if you have unmodified aptamer, especially in the case of RNA. We are using these chemically modified aptamers, which have this fluoropyrimidine modification, and this makes them stable in vivo. Then, because they are very small, they can be filtrated through the kidneys. And to avoid that, you can attach a bulky moiety, for example, PEG, or then you can have multivalent aptamers, which means aptamers which can bind to different targets and which are joined together to form a singular molecule. So by just increasing the size of the aptamer, you can avoid this renal filtration. And then, since these aptamers have not been studied as much as antibody, there is still some questions regarding their toxicity, which of course need to be clarified. So now, if we remember what were the problems with these ocular drug deliveries, that one is that these drugs can't go across ocular barriers. And then the second thing is that they are quickly eliminated from the retina. So that's why we are planning to use these aptamers, not so much as a therapeutic agent, but as an active targeting agent, so that they would increase the probability of redirecting drugs to the retina and also slow down the elimination of the drug. Because if they will bind quite tightly to the retina, hopefully they won't be eliminated so quickly. And as you can see, many different types of molecules have been already linked to aptamers. Here you can have, for example, an siRNA. In violet, you can always see the aptamer. Or you can attach a protein or an antibody. We are also interested in attaching some chemotherapeutic drug, because there are some ocular tumors which are actually child tumors, retinoblastoma, and the way that these chemotherapeutic drugs are currently administered to children is very invasive. So we would like to develop some better ways to administer these drugs so that they would only target these cancer cells. And then, of course, you can make these multivalent aptamers so that you can combine an aptamer which binds a certain target to an aptamer which binds another target. And the idea of doing this in vivo CELEX came when I was reading this paper about this selection which was done for identifying brain targeting aptamers. And here, this A15, it's the targeting aptamer, while this SCAP is this control aptamers. And in this figure, you can see that this targeting aptamer is targeting much more effectively to this parenchyma in the brain compared to the non-targeting aptamer. And here, the choice of the colors, it's a little bit bad, but in blue, you can see the cell nuclei, and in purple, the aptamers. So you can see that here there is mostly blue in the case of the control. You don't see much violet, but then in the case of these aptamers, I can't really see the cursor because I have the photos of people here, but you can see more violet in the case of these aptamer, meaning that it will eventually go to the brain after, I think this was after intravenous injection. Okay, and now here to this method which we are now using. As I mentioned, we are using this RNA library which has 10 to the 15 sequences. And we have 3 different administration routes: topical, subconjunctival, and systemic administration. After this aptamer library is administered to rats, they are left in the rat for half an hour to 1 hour. And then the animals are euthanized, and we isolate the ocular tissues, extract the RNA, amplify, and then pool the RNAs from different ocular tissues and then inject it again to the animal. And we have now done about 10 rounds of selection, and we hope that by 20 rounds we can eventually find some targeting aptamers. Here is a little bit more in detail that how this, for example, how we are evaluating how this aptamer is enriched. Once we have extracted the total RNA from tissue homogenates and done the cDNA, we can then run a PCR with these primers, which are specific for aptamers. So independent of the sequence of the aptamer, all can be detected by the same primers, because the aptamer sequence has 2 constant regions at both ends. And so then you can see, for example, here in the case of retina and retinal pigment epithelium, that in the beginning we don't see the aptamers, but then after about 25 to 30 cycles, we can start to see some aptamers. And we hope that by increasing the selection round, eventually the number of cycles required to find the aptamers will decrease, because we will have more aptamers in these tissues. And this is what we have actually seen, is that this is the topical administration and this is intravenous administration. And on this side, we have the ocular tissues, and on the other side, we have some control tissues, which are non-ocular tissues. And you can see that after round 1, 2, 3, and 4, the number of PCR cycles required to detect the aptamers from ocular tissues is decreasing, while it generally stays quite constant for these non-ocular tissues. And in this way, we could a little bit follow how this enrichment was done even before sequencing the samples. Then this was the samples which were actually sequenced at round 8. This was for intravenous samples. And what we can see from this heat map is that the bars in red indicate that there is aptamer enrichment. And here in pink, you can see control tissues, and in blue, there are these ocular tissues. And here are the different ocular tissues. And you can see that, for example, this one family of aptamers seems to be more enriched in the ocular tissues, while these are more into these non-ocular tissues, which of course is to be expected, especially in the case of intravenous injection. And I think if we compared those earlier rounds to this round number 8, we could see more clearly that there were these kind of families of aptamers which were enriching and which were diverging between these ocular and non-ocular tissues. And this is also this principal component analysis. I haven't done this myself, so this was actually done by Søren at Aarhus University. But what you can see here is this intravenous injection round 4 and then round 8. And you can see that in round 4, ocular and non-ocular tissues were quite clustered together, so you couldn't really see any difference between these aptamers. But then at round 8, we can start to quite clearly see that these ocular tissues, which have this blue center, are different from these non-ocular tissues, which are in red. But these are all relative data, so we don't have any kind of definitive results for these yet. We just can start to see some enrichment of aptamers towards these ocular tissues. And then we have also this kind of a little bit funny results that this retina and lens seem to be binding in a very different way. But I think we shouldn't read much on that. We should just continue with the selections and then see further on when we have more data. So then, in conclusion, what we would like to achieve is to have this local, less invasive ocular delivery strategies for retinal treatment. And we hope to achieve this by using these ocular barrier penetrating aptamers, which would provide active drug targeting, which means that the drug will be basically transported by these aptamers to the correct location inside the eye. And if this can be achieved, it could be a major improvement compared to current intravitreal delivery method. What we can see from our data, and of course these are preliminary data, is that we can see some families which are enriching to ocular tissues compared to the control tissues. But however, we need to, of course, continue with these selections, and hopefully some specific aptamers will emerge at later selection rounds. And once we have selected these aptamers, of course, we would like to attach some cargos to them. We are using this kind of strategy that we have one-fits-all LNA or locked nucleic acid oligolinker. This linker binds to this constant region of the aptamer. That's why it's one-fits-all. And you can then bind your drug of interest, for example, an IgG or some chemotherapeutic drug, to the oligo, and then eventually this will hybridize to the aptamer. We are also interested in producing bispecific aptamers or some hybrids. And then eventually, to again improve these patients' well-being, it would be nice to have some kind of targeting with a controlled release system. So we would like to embed these aptamer conjugates into some hydrogels, which could be, for example, injected in the subconjunctival space, and which would then release these aptamer drug conjugates over a long period of time. So this will decrease the administration frequency of these drugs, so that hopefully we can have an administration of once every 6 months or so. So then with that, I would like to thank, of course, the people who did the work and helped us here at UEF. I would like to thank my students, Katja and Sonja. They did all the selections. And then Elisa Torropainen is helping us with the animal studies, and Kati Sisko-Vellonen is doing the mass analysis, mass spectrometric analysis. And then our collaborators at Aarhus University, where I was doing my postdoc and where I actually learned this, this method. Professor Jörgen Kiems, Dr. Daniel Dupont, and Søren Fjeldstrup. And then, of course, the funding, which have allowed us to do this research. And I would also like to thank you for your attention.